AP 120, Lab 3. The topics covered DNA replication and transcription. So, what is this? I hope you automatically know that is DNA, the double helix of DNA. So, DNA is a lovely double-stranded nucleic acid. Its official name, deoxyribonucleic acid. And if you look at it, you can see that on the outside, the strip is the sugar phosphate backbones. The outside strip or pole is the sugar phosphate backbone, and then on the inside is where we find the bases, the nitrogenous bases, hydrogen bonding to each other. There's also a ribonucleic acid, RNA. RNA is a single-stranded nucleic acid, although it can form more complicated three-dimensional structures by having base pairing occur within the strand. So, DNA is the genetic information. It's broken up into about 3,000 genes on 23 pairs of chromosomes. That's right, most human beings have 46 chromosomes in total. And each gene codes for a specific protein. So here we have the cell membrane, here is the nucleus, and then the DNA. The DNA, of course, broken up into those 30,000 genes. Each gene will occasionally get an RNA copy produced that will then leave the nucleus. And of course, when cell division occurs, the DNA can condense down into chromosomes. So, nucleotides are the monomers for nucleic acids. And a nucleotide has a phosphate group, a 5-carbon sugar, and a base. So the monomer for nucleic acids is a nucleotide, which has a phosphate group, a 5-carbon sugar, and a base. And what happens to form the long strand is you get covalent base pairing between the sugar and the phosphate to give you that long strand of RNA or the long strand of DNA. The DNA, of course, has 2 long strands. All right, so here is again a close-up of a nucleotide. Again, phosphate group, sugar, nitrogenous base. Phosphate group, sugar, base. That is what makes up the nucleotide. And the sugar is slightly different between RNA and DNA. It's ribose sugar in RNA, deoxyribose in DNA. The bases, it turns out, there are 5 different nitrogenous bases. There's cytosine, C, thymine or T, uracil or U, adenine or A, guanine or G. So you need to know the names of the 5 bases. You don't need to know their structure. All right, deoxyribonucleic acid is found in the nucleus, although there is some in the mitochondria. DNA has deoxyribose sugar, the phosphate group, of course, and 4 other nitrogenous bases. Adenine, A, guanine, G, cytosine, C, or thymine, T. A, G, C, T are the types of bases in DNA. No uracil, no U. So here we see it. Here's the sugar phosphate backbone. Here are the bases, T's, G's, A's, no U's. All right, the double strand, of course, is held together by hydrogen bonding between the complementary bases. And it turns out there are specific bases that hydrogen bond with specific other bases. So for complementary base pairing, thymine always pairs with adenine, and adenine with thymine, so T to A or A to T. And guanine always pairs with cytosine, or cytosine to guanine, G to C or C to G. So here's an A pairing up with a T. Here's a C pairing up with a G. Now, what is a hydrogen bond? Well, it should be pretty obvious looking through these options. It's the one with the word hydrogen in it. There's an attraction between slightly positive hydrogen and slightly negative nitrogen or oxygen. All right, RNA. RNA is single-stranded, although it can form more complicated structures within itself. It is found in the nucleus and within the cytoplasm, so both in the nucleus and the cytoplasm of the cell. RNA has the ribose sugar, phosphate, of course, and 4 nitrogenous bases. Adenine A, guanine G, cytosine C, or uracil U. Now the basic complementary pairing is the same, except we just switch out uracil for thiamine. So in RNA, A pairs to U or U to A, and C pairs to G or G to C. Now RNA is more functionally active, so there are three different kinds of RNA. You have messenger RNA, or mRNA, ribosomal RNA, or rRNA, and transfer RNA, or tRNA. Now, here are three sequences of nucleotides. Which of these sequences come from DNA, which come from RNA? That is right. This one comes from DNA. Look at that T. It's a clue. It's the DNA. These two come from RNA. Look at those U's. Those are clues. These are RNA. What about complementary base pairing? Figure out what the complementary bases are for each of these nucleotides. Pause the video if you feel you need a little more time. Hopefully you got the correct answers. A pairs to T, T to A, C to G, C to G, G to C, A to T, U to A to U, A to U, A to U, G to C, U to A, and so on. All right, DNA has two big jobs. First is to be able to make a copy of itself, because we need a brand new copy of DNA for cell division in order to have a complete set of DNA in each of the new cells. And this is called DNA replication. DNA replication, sometimes known as DNA synthesis, is producing a new copy of DNA. Now, what do you need to do this? Well, you need the original DNA that is required. You need DNA nucleotides. Again, nucleotides, the monomers for making DNA, and of course it's deoxyribose nucleotides. You need an enzyme complex called the DNA polymerase. This enzyme is what makes the DNA chain. It brings in the complementary DNA nucleotide and then attaches it to the growing chain. DNA polymerase makes the new DNA. You also need helicase. Helicase is an enzyme that opens up the double helix of the starting DNA. And there's actually a lot of other players involved too, but we're not going to worry about those. So DNA, DNA nucleotides, DNA polymerase, and helicase. So we look at this picture here. We see they have the original DNA. The original DNA is being opened up by the helicase enzyme to give us two strands. And then DNA polymerase is attaching to those strands and making new DNA. So the DNA polymerase brings in a new nucleotide that complementary base pairs with the nucleotide's base. That's part of the original DNA. And then it just keeps growing and growing, adding more and more nucleotides until it completes the production of the new DNA molecule. And on the other strand, it basically does the same thing. It's a little more complicated, but we're not going to worry about that. So DNA polymerase attaches to the single-stranded DNA, brings in DNA nucleotides that complementary base pair with the base of the DNA strand, and then attaches it to the growing chain of DNA to give us a new DNA molecule. All right, the other big job for DNA is transcription. Transcription is when the specific instructions of a gene, and just a gene, so just a portion of the DNA, needs to be turned into messenger RNA so that that information can leave the nucleus and go into the cytoplasm. So we're making a messenger RNA. We're making the code, the sequence for one gene and only one gene. To do this, we need, of course, the DNA, because that's where the gene is. That's where the information is. We need RNA nucleotides. Again, nucleotides are the monomers. We need ribonucleotides because we're making RNA. And we need the RNA polymerase. RNA polymerase is the enzyme complex that opens up the DNA double helix, opens the DNA up, and makes the RNA chain. So RNA polymerases make RNA. And here's a picture of how this would work. Here is the double-stranded DNA. The RNA polymerase attaches to the DNA at the beginning of a gene, opens up the double helix, and then it just brings in an ribonucleotide that complementary basis pairs with the sequence on the DNA, and then just attaches it to the growing chain to form the messenger RNA. So here we see A to U, G to C, C to G, U to A, bringing in the ribonucleotide, the RNA nucleotide, that complementary base pairs with the sequence on the DNA. And the DNA, as we know, stays in the nucleus where it's safe. We want to keep our DNA protected. It has the information that makes us us, that allows the production of all the things needed for us to function. So when we need to make proteins, we send out the messenger RNA. So the messenger RNA will then leave the nucleus, go into the cytoplasm. Things are more dangerous, and there get used to make the proteins. And that is it for this part of Lab 3.